In step 2 of the upload process, we need to describe the microbial strains that the study examines. We can start typing a microbial strain in this input. For example, Roseburia intestinalis. As you can see, as I type, this list is going to update with the different strains. I can type Bacteroides, and you can see that even before finishing typing, I get some results. I can type Bacteroides theta Omicron. I don't have to type the entire thing. I can just pick these. Every time I add a new strain here, it gets added to the list with a link to NCBI so that I can validate whether or not this is the actual strain that I am looking at. Right now, these taxa are at the species level, but we can also pick strain level, and this is recommended, ideally, to be as precise as possible about what exact strains we are using in our experiments. I can click on the little X's to remove these records and try to find more precise taxa. For example, Roseburia intestinalis DSM. Unfortunately, I can't really find any strain that matches this DSM identifier. The data we're looking at is coming from NCBI. If you look at their taxonomy browser, you can see that these are all the strains that they have on Roseburia intestinalis. Now, the particular strain I might be looking for is DSM 14610. However, as you can see in BacDive, this strain is present here, but it is not present in NCBI. However, this strain designation, the canonical strain designation, according to BacDive and according to NCBI, is L182. In fact, we can click on this and find this specific strain. So what I can type here is type L1, and you can see that this is a strain that we get. Another thing to note is that if we start typing a word that is inside of a particular strain, for example, theta iota omicron, it will not be found. While the matching in the search is partial, bear in mind that it always needs to match the beginning of the strain. So you do need to either paste the strain designation or a species, or start from the genus designation. So I can type Bacteroides, and that will find something, and then type VPI, Bacteroides theta Omicron VPI, and you can see the highlighting matches what I've typed right here, but it has to match the beginning of the string. The reason for this is technical. This is a large database of a lot of strains, and so it is difficult to match them otherwise. But hopefully this should be sufficient to find the specific strain that you're looking for. Now, let's say that there's a particular strain that is not present here, and we can't really find... Now, let's say that there's a custom strain that we've developed in our lab, maybe with some specific mutation, maybe it's adapted to work in a specific medium. What we can do is we can click on the Add Custom Strain button. By doing this, we get this form, which allows us to describe a specific strain that is connected to a parent strain. For example, I can type Blautia hydrogenotrophica, and again we get the same sort of completion. We do not have to type the whole thing. We can pick a species, for example, or maybe we started with a particular strain. Ideally, we want to be as precise as possible, and by doing this, the name is going to be pre-filled, but instead of, let's say, DSM 10501, we can say something like strain, custom strain 001, or something like this. And in the description here, we can describe what it is that we mutated, or what precise technique that we used. We can add another custom strain. We can click on the duplicate button to get another strain that is duplicated with the same parent species and the same sort of microbial strain. This can be useful to sort of shortcut the process of finding the parent. Bear in mind that these strains do need to be unique. So if I try to submit both of these with the same name, and I click add custom strain to add another one, this will get validated and we'll get a message that says strain names are not unique. We do need to somehow modify these, or this could be a good check to point out that maybe we made a mistake, we've accidentally added a particular strain twice. And now if I add custom strain, I can actually add another one, a third one, or a fourth one, and I can click on the remove button to remove this form. If I'm happy with this state of things, with this list of strains, we now have this strain, these two strains that come from NCBI, and this custom strain. I can click on the next button to finalize this step. And now we get to step 3, measurement methods, which we're going to take care of in the next video. If there's a particular strain that you think should be added to the system, we can do this manually. All you need to do is reach out to us. You can click on the About page and find the contact details of the Lab of Microbial Systems Biology. You can scroll down and find our website or a link to send us an email, or you can open a discussion on GitHub. Either way, please reach out and we might be able to accommodate your situation in order to ensure that this data is as accurately described as possible.